ලංකාවට අපි හඳුන්වා දෙන්න යන්නේ මේ කියා ස්ටාර්ට් කියන්නේ ඩෙන්ගු ටෙස්ට් කිට් එකක්. දැන් මෙච්චර කාල තිබුණ ටෙස්ට් කිට් වලින් ප්‍රයිමරි ඉන්ෆෙක්ෂන් උයි සෙකන්ඩරි ඉන්ෆෙක්ෂන් කියන දෙකම එකවර හඳුනා ගන්න අනිවාර්යයෙන්ම හොස්පිටල් එකට යන්න ඕනේ සාම්පල් එකක් දෙන්න ඒකේ රිසල්ට් එක ගන්න. හැබැයි දැන් අපිට තියෙනවා මේ එක සිම්පල් ටෙස්ට් එකකින් මිනිත්තු 20ක් ඇතුළත අපිට දැනගන්න පුළුවන් අපිට තියෙන ප්‍රයිමරි ඉන්ෆෙක්ෂන් එකක්ද සෙකන්ඩරි ඉන්ෆෙක්ෂන් එකක්ද මික්ස්ඩ් එකක්ද කියන එක. මොකද මේ කැසට් එකේ තියෙන මේ වින්ඩෝස් දෙක තියෙනවා. ඒ ටෙස්ට් දෙකම මේ එක ටෙස්ට් එකකින් කරනවා. එතකොට එතන මොකද්ද එතනින් ජනතාවට වෙන හොඳ? ඒ කියන්නේ ප්‍රයිමරි ඉන්ෆෙක්ෂන් එකයි සෙකන්ඩරි ඉන්ෆෙක්ෂන් එකක් දෙකක් තියෙනවනේ දෙකම ඩිටෙක්ට් වුණාම එතකොට එයාට ඉමීඩියට්ලි ඒකට අවශ්‍ය ඒ කියන්නේ මැනේජ්මන්ට් එක හොස්පිටල් එකට ඇතුල් වෙලා කරන්න පුළුවන්. එතකොට ඒකෙන් ඒ ඩෙන්ගු වල තියෙන ඩෙන්ගු වලින් කෙනෙක්ව බේර ගන්න තියෙන ලොකුම දේ තමයි ඒක පුළුවන් තරම් ඉක්මනට අපි දැනගන්න ඕනේ අපිට ඩෙන්ගු තියෙනවද කියලා. හැබැයි දැන් අපිට ඩෙන්ගු තියෙනවද කියලා විතරක් නෙමෙයි අපිට ක්ෂණිකව දැනගන්න පුළුවන් විනාඩි 20කින් අපිට තියෙන ඩෙන්ගු ප්‍රයිමරි ඉන්ෆෙක්ෂන් එකක්ද සෙකන්ඩරි ඉන්ෆෙක්ෂන් එකක්ද මික්ස්ඩ් ඉන්ෆෙක්ෂන් එකක්ද කියන එක අපිට විනාඩි 20ක් ඇතුළත දැනගන්න පුළුවන්. දැන් ඒ හැකියාව අපි ලංකාවට ගෙනල්ලා තියෙන්නේ මේ කියා ස්ටාර්ට් කියන ඩෙන්ගු කිට් එක ලංකාවට ගෙනල්ලා. දැන් මේක අපට දැන් ජනතාවට මේක මේ කිට් එකේ තියෙන බෙනිෆිට් එක ගන්න නම් ඒගොල්ලෝ යන්න ඕනේ ඒගොල්ලන්ගේ ડોක්ටර්ස් ළඟ. මේක ෆාමසි වල ගන්න තියෙන්නේ නැහැ. මේක තියෙන්නේ ડોක්ටර්ස් ළඟ. මොකද ડોක්ටර් කෙනෙක්ගේ සුපර්විෂන් එක යටතේ මේ ටෙස්ට් එක කරන්නේ. ඉතින් දැන් ජනතාවට GPsලා ඒගොල්ලන්ගේ ලෝකල් GP මාර්ගයෙන් තව ඉස්සරහට ආණ්ඩුවේ ස්පිතාල වලත් tie කියලා අපි බලාපොරොත්තු වෙනවා. එතකොට විනාඩි 20කින් ඒගොල්ලන්ට ඒක දැනගන්න පුළුවන් ඩෙන්ගු තියෙනවද නැද්ද කියලා. ඊට පස්සේ ඒකට අවශ්‍ය ප්‍රතිකර්ම ටික වලට ඒගොල්ලන්ට යන්න පුළුවන්.
Dengue combo kit, we have very sensitive detection of immunoglobulin G, so-called IgG. By the detection of very low concentration of IgG, we can distinguish whether it comes from the primary infection of dengue or secondary infection of dengue. By differentiation of primary and secondary infection of dengue, we can save a lot of the people. In other words, we can reduce the mortality of the people from the dengue infections. The reason is we have very low detection limit, LOD, limit of detection, because we have some proprietary technology to detect the IgG from the dengue. We can make the differentiation between primary infection and secondary infection. That is the story of tonight.
Life is precious regardless of nationality, race, or religion. Founded in 2002, Access Bio is committed to saving lives and instilling hope around the world through accurate, early diagnosis of diseases. We have built up an extensive customer network, including various international organizations such as the WHO, the Global Fund, and Médecins Sans Frontières. In March 2016, Access Bio successfully acquired an investment of $8 million with the Global Health Investment Fund, GHIF, supported by the Bill and Melinda Gates Foundation. We believe this collaboration will serve as a stepping stone for us to further contribute to the improvement of global health. Since our listing in the Korean Stock Exchange in May of 2013, we have expanded our global operations in Korea and Ethiopia. Access Bio, in collaboration with Wells Bio and Medisensor, strives to maximize its synergistic effect to reinforce its global business capacity and expand product lines through effective use of manufacturing and R&D resources. As the trends of in vitro diagnostics industry are shifting from treatment-centered to preventative early diagnosis for timely treatment, the demand for point-of-care tests with enhanced sensitivity, convenience, and competitive prices continues to grow. To lead the diagnostic markets, Access Bio is pioneering highly sensitive next-generation products with our core technologies, incorporating immunochemical, biochemical and molecular diagnostics. Access Bio's malaria RDTs have been supplied to many corners of the world and have achieved more than 60% of market share, solidifying our leading position in the global market. Utilizing HST technology, our products were evaluated as top performers for their unparalleled quality and high sensitivity in the WHO performance tests. In addition, we have upgraded our immunochemical diagnostic platform with newly developed Ultra HST technology that has enhanced sensitivity by more than 10 times compared to the current technology. Access Bio continues to make inroads into the immunochemical diagnostic market with cutting-edge HST-TRF and HST-S technologies. In the near future, we will offer an immuno-POCT, integrating our biosensor technology that utilizes biotechnology, information technology, and nanotechnology for significantly improved performance. In the field of biochemical diagnostics, Access Bio has developed the world's first true point-of-care G6PD screening test, which is a major driving force for our ever-growing company. G6PD deficiency is a genetic disorder that affects around 350 million people around the world, accounting for about 5% of the world population. G6PD deficiency causes the destruction of red blood cells and acute hemolytic anemia, resulting from taking certain foods or drugs with high oxidative stress, such as vitamin C, painkillers, and anti-malarial medicines such as Primaquine. Therefore, the WHO has recommended G6PD deficiency screening tests for safe use of Primaquine and officially recognized Access Bio's G6PD RDT as the only commercially available test that can be performed on-site in tropical settings. With our advanced biosensor technology, Access Bio will continue to take the leading position in diagnosis of not only G6PD, but also many other related diseases by introducing an all-in-one biosensor that simultaneously detects at least 10 different diseases in just a few minutes. In the field of molecular diagnostics, Access Bio has developed and patented its SBS primer platform technology that allows multiplexing assays with enhanced sensitivity and specificity. Based on the unique multiplexing technology, we are making steady progress in the development and commercialization of various molecular diagnostic tests such as HPV, TB, and the Zika virus. Our HPV tests can be customized to meet the diverse needs of both emerging and developed markets, thanks to their outstanding performance and competitive price. Aiming for diversity in high-value private markets, Access Bio has expanded its product portfolio to include products popular in developed countries, such as respiratory disease diagnostics and HbA1c biosensor. The respiratory disease diagnostic market is rapidly growing due to an increasingly older population and severe air pollution. Our newly developed flu A and B test has successfully improved sensitivity by 10 to 100 times compared to its previous iteration. Strep A and RSV will incorporate the same platform technology to strengthen our respiratory disease diagnostic product portfolio. Access Bio's glycated hemoglobin biosensor is a fully automated one-step sensor which provides an accurate diagnosis of diabetes. The convenience and high accuracy of the biosensor makes it a very competitive product in the market. In the near future, this biosensor will be a multi-platform diagnostic tool that uses the same one-step process to diagnose heart disease and other related illnesses. Through product portfolio differentiation and expansion, Access Bio is working to penetrate developed markets. Access Bio's R&D has developed our technologies from immunochemical to molecular diagnostics, and we are expanding our product pipeline to include the diagnosis of not only infectious diseases, but also hereditary and veterinary diseases. With our innovative technologies, technical capabilities, and global network, Access Bio will continue to leap forward to become a global top 10 in vitro diagnostic company. Believing the basic principles of cherishing life and health for all, Access Bio will continue its dedication to the prevention and early diagnosis of diseases and help save lives across the globe.